All right. Hello, everyone. My name is Stoutjesdyk, and I am group number one. So I did a presentation on teratogens and their effects on a developing embryo. So teratogens are drugs or any other substance that are used by a pregnant woman that have a physical or functional effect on the developing embryo. This can include anything from illicit drugs and alcohol to prescription medication, even food and clothing dyes, or any substance in the environment that can affect a embryo. Most teratogens have a specific time and dose-specific effects on developing embryos. And this means that depending on how much exposure occurs and during what stage of pregnancies, many teratogens can cause different developmental issues for the embryo. Historically, new teratogens were found when birth defects became more prevalent, and then researchers would backtrack and try to figure out what caused those birth defects. However, in more recent years, there's been a higher focus on studying possible teratogens before they can cause birth defects. And one of these studies was conducted by the researchers from Tam Kang University in Taiwan. They wanted to look at topiramate, and then to do this, they used a zebrafish as their model organism. And despite the fact that zebrafish are obviously very different from humans, they are a surprisingly good model organism for humans, especially for their development. And they share about 70% of their genes with humans. And they're also much cheaper, require less space, and produce faster. They reproduce faster and in larger quantities than mice do. So they wanted to look at topiramate, and topiramate is a drug that is commonly used to treat epilepsy in adults. And epilepsy is one of the most common neurological disorders in the US. So looking into the teratogenic effects of this drug is very valuable research. So the FDA classifies topiramate as a pregnancy category D drug, which indicates that there has been possible evidence of fetal risk in humans through previous studies, but also there's a potential benefits from the use of the drug for the pregnant women themselves. Excuse me. So also through other studies, there has been proposed to be a connection between topiramate use in pregnant women in the first trimester of pregnancy and the development of oral clefts among their newborns. So these researchers decided that they wanted to see if those claims were legitimate or not using zebrafish. To do this, they treated a total of 82 fish with topiramate over the course of 7 days. The average human dose for topiramate is 500 milligrams per kilogram per day. And in order to scale this down, researchers fed the fish 0.5 milligrams per gram per day of a topiramate blended dry food. At the end of the final day, they bred the zebrafish and then collected the embryos the following morning. They did find that only about 40% of the fish were able to lay eggs. And then the researchers wanted to look at a few different factors that were affecting the embryos from these eggs. So first, they looked at the weight of the offspring. One of the main side effects of topiramate in humans is loss of appetite and weight loss. And to test this, they wanted to see if this was also the effect in zebrafish as well as humans. So they found that the average weight of the experimental group increased by 0.08 grams throughout the feeding process, and it only increased to 0.2 grams for the control group. They found that this was not a big enough difference to have any sort of statistical significance when comparing the two groups, and therefore they found that this is not an effect that topiramate has on zebrafish like it does in humans. They also wanted to look at the maturation of oogenesis. So after the embryos, after the zebrafish were treated with the topiramate, they were then sacrificed, and their ovaries were collected to observe the ability to obtain, the ability to perform oogenesis. And a substantial portion of the ovarian tissue was filled with the mature oocytes in the control group. However, they found that there was a significant decrease in the percentage of mature oocytes that was detected in the treated females. And this indicates that the exposure to topiramate in the female fish has a negative effect on the oogenesis. They also wanted to look at the epiboly progression in the offspring fish. They found that the survival rate of the embryos after 8 hours of fertilization from the treated group was lower than the control group. And the researchers determined that this was due to the treated cell's inability to perform epiboly correctly. And to ensure that this was in fact caused by the treatment of topiramate, the researchers then microinjected the embryos with 3 different dosages of topiramate during the 1 and 4 cell stages. They then observed the survival rate of these embryos after 8 hours, and they not only found that topiramate itself had a much higher mortality rate than the control group, but they also found that this had a dosage dependent reaction. So the higher the dosage of the topiramate, the lower the survival rate was of these embryos 8 hours after fertilization. They also wanted to look at the cartilage development of the offspring. Cartilage is the predominant component of the cranium in early embryogenesis of zebrafish, and it becomes fully developed 74 hours, about 3 days after fertilization. So after 4 days of fertilization, the researchers observed the cartilage structures of the offspring fish. And in doing so, they observed that about 23% of the offspring zebrafish that were treated showed cartilage abnormalities. These abnormalities included a malformation of Meckel cartilage, a malformation of ceratohydal cartilage, and also of the ethmoid plate during embryogenesis. And they also found that there was a severely underdeveloped ceratobrachial cartilage. Finally, they wanted to look at craniofacial malfunctions. And to do this, the researchers measured the ratio of the length and width of the cranium region of the treated motherfish. And the treated fish showed significantly decreased ratio between the length and the width within the craniofacial region compared to the control group. Additionally, the number of spinal segments was also counted, and on average, 13.2 spinal columns were developed in the control group, while only 6.2 developed in the treated offspring. And these results suggest that topiramate not only can be linked to malfunction in cartilage development, but also impair bone formation itself. So the study itself was designed and conducted in a way that aligns with the scientific method. There was a single variable that was changed throughout the experiment, whether or not the group of fish would receive a dose of topiramate or not. And the researchers looked at many different potential effects that the drug might have on the fish. In doing so, they were able to provide a wide range of data pertaining to the effects of this drug on development. However, on the other hand, this did mean that the researchers were not able to focus on one aspect and expand more on that specific development malfunction. The paper itself was not organized in a traditional format. The researchers reported the methods and results for each side effect observed separately from one another, instead of making a singular method section and a singular results section. This made the paper hard to read, and then also it felt really unorganized. So drugs are not the only teratogens that pose risk to a developing fetus. Any substance in the environment can be a factor. So these researchers in Cairo University in Giza, Egypt, they wanted to look at taurinazine, or TAZ for short. And this is one of the most commonly used artificial dyes for foods and drugs to achieve a yellow color. So to observe the potential teratogenic effects of TAZ, they used Wistar rats. And to do this, the rats were bred, and then the pregnant rats were divided into 3 groups of 30 rats each. One received a lower dose of TAZ at 0.45 milligrams per kilogram. One received a higher dose of TAZ at 4.5 milligrams per kilogram. And then the third group was used as a control, and they only received saline treatments. So first they wanted to look at fetal resorption and mortality rates. So they found that in the group that was treated with a lower dose of TAZ had 6 reabsorbed fetuses in the group, and then also a 7.1 mortality rate. And also in the group that had a higher rate of TAZ found that they had 10 reabsorbed fetuses in the group, and then also a 10.5 mortality rate. So they determined that this was not significant between the 2 doses. So this response does not have a dose dependent, but also they found that the control group had much lower results than this. So it was significant compared to the control group, just not controlled to each different dose. They also looked at the fetal weight and length, and they found that the weight and length in both of the groups that were treated with TAZ were found to be significantly smaller in the fetuses in the control group. And you can see a really good picture of that. The larger fetus is the control group, and the smaller fetus is a fetus in the TAZ treated groups. So researchers also wanted to look into the organs and skeletal systems and see how TAZ affected these as well. They found that the fetuses treated with the lower dose of TAZ showed signs of cardiomegaly and liver damage. And then the fetuses in both of the groups treated with TAZ showed signs of hemosiderin in the spleen, and then also necrotic renal tubes. So they also stained and looked at the development of the skeletal system as well. And they found that the fetuses that were treated with TAZ showed skeletal malformations, including missing coccygeal vertebrae, missing sternal vertebrae, missing hind limbs, and irregular ribs. So according to the FDA, the acceptable daily intake of TAZ is 3.75 milligrams per kilogram. However, if these researchers' data are correct, they found that there is a significant decrease in weight and length of a fetus, and also an increase in skeletal malformations in the fetus that were treated with only a 0.45 milligrams per kilogram per day dose, which is obviously much smaller than the recommended 3.75 milligrams per kilogram. Another thing to keep in mind is that TAZ is one of the most popular dyes in food, medicine, cosmetics, clothing, etc. And so it's very possible for people, including pregnant women, to be exposed to it from multiple different sources in one day. And that's something that they need to keep in mind. So the research itself was conducted in a professional and scientific manner. Their experimental design was done well for what they were looking to test. There was only one variable that was changed throughout the experiment, and this was the dosage of TAZ that was given to each group of rats. This also ensures that any results that are observed during the study had a clear cause. And the researchers were very consistent in the method that they bred each rat, that they administered the TAZ, and that the rats were taken care of after the study, during the course of the study, and then also after the study as well. The paper itself was laid out in into logical sections that were easy to read and follow, and it also followed the sequence in chronological order, which was very helpful to understand. All right, and here are some of my references, and thank you guys so much.